A similar observation is made when you inhibit another inhibitor. If you inhibit this LINGO-1, and again, this is an animal model, and on your left, you see the normal spinal cord. In the middle, you see a spinal cord that is in an untreated animal with this MS-like disease. And on your right, you see an animal that has been given an antibody that counteracts the effects of the inhibitor. And if you look at the sites where the arrows are, you can see that the normal animal has a lot of blue, that's the myelin. The animal in the middle has lost its myelin, but if you inject an antibody to this LINGO, to this inhibitor of oligodendrocyte maturation, what you see is remyelination. So these are the first observations, really, I think, very exciting, that have shown the ability to inhibit inhibitors that are there normally and preventing the brain from perhaps healing itself. Very exciting. Of course, the issue is now how can we translate these findings into people? And that's going to be a major hurdle. Now, what does this mean, though, in terms of stem cells? Everyone has been asking about stem cells. But before we even talk about them, I think what these observations really point out, very importantly, is that the precursors of healing are there. They may not be limiting. You have to control these native inhibitors of remyelination, and that you may not even need stem cells, because, of course, the purpose of stem cells would be to regenerate, if you will, cells that have the capacity to remyelinate. They're already there. All you have to do is allow them to do their own thing. And stem cells, of course, have their own particular issues. How do you get them into the brain? And once they're in the brain, how do you control them? How do you keep them from just multiplying and becoming a tumor? So these are all very important potential side effects of having stem cells. But what about stem cells? Well, there are two ways that they have a role to play in MS. One is they can be used to regenerate cells of the central nervous system, oligodendrocytes or nerve cells, or as they're now being used, they're being used to regenerate cells of the bone marrow. You've heard about bone marrow transplantation for MS, and people with very severe illness have been treated in this fashion. They've had their bone marrow essentially wiped out, reconstituted with stem cells in the hope that this new immune system, if you will, is going to be less harmful. Now, the data on bone marrow transplant is not in quite yet. It turns out that it may not be as effective as had originally hoped for reasons that I can talk about later. But what about stem cells in terms of their potential value in a whole different context? Well, as are shown to you here, MSC is mesenchymal stem cell, and they can differentiate into any one of a number of different cells based upon how you culture them, how you expose them to different cytokines or nutrients, if you will. They may be difficult to control once they begin to proliferate, but there have been experiments done in animals, again, with this EAE, this MS-like disease, that have been injected with stem cells, the hope being that the stem cells would enter the brain and repair areas that had been destroyed. What turned out was that if you injected stem cells into the brains of animals with this MS-like disease, they indeed got better. But the reasons for their improvement were a total surprise. The reasons that animals improved after having an injection of stem cells was not because the stem cells regenerated into brain cells, but because they inhibited the immune response. So this is shown to you schematically here, showing you that these mesenchymal stem cells have the ability to decrease the inflammation that occurs in these animals with an MS-like illness. So a totally unexpected mechanism of action, a mechanism of action that was opposite of what originally had been thought, but one that was nevertheless effective. So these Stem cells are acting as immune modulators, as immune suppressants, if you will. How they do this is unclear at the present time. That doesn't mean that they might not be valuable in MS, but if they don't regenerate tissues, there might be other ways, perhaps, of modulating the immune system. And of course, in the brain, you can see the same kind of reaction. They do have a protective effect, but they do so by preventing damage rather than regeneration. Damage to nerve cells, damage to nerve cell precursors, and of course to oligodendrocytes. And they also inhibit the activities of two very important populations of cells, the so-called microglial cells, or macrophages, and astrocytes. So yes, there's a role for stem cells, but an unexpected one, and one that I think is very different than had originally been anticipated. Now let's talk about some of the disease processes in MS. When I went to medical school, I was told MS was an acute inflammation of the brain that affected mainly the white matter, the myelin. And indeed, that's the most prominent component of the disease early on. When one has attacks, when one has relapses or exacerbations, one sees these hot spots, if you will, these areas of active inflammation on MRI scan, and we assume that those are the ones that are causing the various difficulties. This inflammation, as you see, occurs in patches. It's multifocal. It's mediated by certain cells of the immune system, namely lymphocytes and antibodies. It results in a destruction of tissue, of both axons and of myelin. It has the potential to heal, as I talked about. There can be some remyelination, if you will, and it involves mainly the white matter. And we have medicines now. In the past decade, we have medicines that can affect this acute aspect of the disease. We have the beta interferons, and we have glatiramer or acetate. But there's more to MS than that, and this has really only been observed, I would say, in the past 4 or 5 years, though it's been known actually for about 100 years, but really never investigated, and that is the degenerative aspects of the disease. We know that about half of individuals who present with a relapsing remitting course change over a period of time. They don't have attacks anymore, they don't have exacerbations, they just have a gradual progression of their difficulties. And we call this secondary progressive MS. They don't have attacks anymore, their MRI changes. They don't have new lesions, they don't have these acute areas of inflammation anymore. They just have a gradual loss of tissue. And it's very indolent. It can go on for years and years and years. It's diffuse. You don't see it on MRI scan. You actually don't have an idea of this diffuse inflammation that's going on even in what looks to be normal appearing white matter. It's caused by a different population of cells than those that cause the active inflammation. It's caused by these macrophages, these microglial cells, these glial cells, and different compounds are being produced rather than antibodies and various toxic cytokines. It results in a loss of brain tissue called brain atrophy, and what's very, very frustrating for me and for people who have this phase of the disease, there's no available treatment. So this is a whole area that needs to be addressed, that has not been addressed for various reasons, and I can talk about that in a minute. But there may even be a third disease process going on now. And this is very, very new data, very exciting data, because what it tells us is that the kind of MS a person has at the beginning of their illness may be very different 15, 20, 30 years down the road. It's sort of a moving target. Things change, things evolve. One sort of leads to another, we believe, so that a cure for one phase of the disease may not be sufficient in terms of the other phases.